All right, here's our video for Wednesday that will take us through the remainder of Chapter 11. So the remaining part of Chapter 11 is about the mechanisms influencing the human genome throughout the history of our species. And so I titled this, or the name of the file is Evolutionary Genetics. But yeah, all of Chapter 11 is still on population genetics, and what you'll find is a lot of these mechanisms you're familiar with, and it's a good reminder that populations evolve. The allele frequencies change within populations due to these mechanisms, and keeps that as the focus of this section on evolutionary genetics. That is, it keeps our focus on the population. All right, so some key terms. First one, directional selection. And so in directional selection, you have, one of the extremes selected for. And so you're either increasing in allele frequency, or you're decreasing in allele frequency, or you're increasing in the phenotypic expression of that. And so a good example of humans is going to be the increase in melanin production as a result of high exposure to sunlight. And so there you can see evidences in certain people groups of increased selection for melanin production, resulting in an increase overall in melanin production. Now, another good example is the decrease in melanin production you have in even some of these populations that are now moving to areas where sunlight is less direct, and where you get a lower amount of it, fewer days where you get good ample sunlight, as you're actually seeing directional selection for less melanin production because of vitamin D deficiency, providing some of that selective pressure. Stabilizing selection. So stabilizing selection is where you're selecting against both extremes, and you are increasing the proportion of individuals that have whatever that average phenotype is. And so here you can see examples of this where being too tall or being too short might be selected against, but melanin production is probably a good example of stabilizing selection overall globally, when you consider that there are basically two balancing forces. One is having enough melanin production to avoid the mutations that come with increased exposure to UV, and then you have the vitamin D deficiency that comes with not getting enough sunlight. Purifying selection. Purifying selection is basically we start to get rid of certain alleles entirely, and just basically maintain a certain combination of alleles. And then balancing selection, where you're not selecting for a specific genotype, but instead you are maintaining a certain frequency of multiple phenotypes. And this is often due to heterozygote advantage, where, typically in our other selective mechanisms, we're looking at one genotype increasing in frequency at the expense of all other genotypes. But in balancing selection, that's not what happens. We maintain a certain amount of certain amount of each genotype, and again, a common reason for that is if individuals that have one copy of each of those two alleles, and they are a heterozygous individual, they have some advantage over either homozygous individual. So the textbook example for this is sickle cell anemia, and how sickle cell trait it provides a an amount of protection against contracting malaria, and it some of the numbers that you find is that about 50% of mild malaria cases are going to be avoided in people with sickle cell trait, that is one mutant allele. About anywhere from 75 to 80% of your hospitalizations are going to be avoided in people with sickle cell trait, and about 90% of very severe cases of malaria that would often result in death are going to be avoided by people with sickle cell trait. And so you can see this heterozygous advantage actually prevents natural selection ridding the population of these lethal alleles. Horizontal gene transfer, this is a mechanism that we see a lot in prokaryotes with transformation, conjugation, even transduction, where viruses are going to transfer genetic material from one bacterium to another. But we're starting to see increased evidence of this in humans. It looks like about 8% of the human genome is the result of retroviruses, and a decent percentage of the human genome looks like it came from our symbiotic bacteria. And so we're starting to see, again, increased examples of horizontal gene transfer. Genetic drift, these are random genetic changes. And so the mechanism underlying the random genetic changes can really be anything, but these are genetic changes that are not the result of some genotype having an advantage over another. And so you get, some examples of genetic drift that are often thought of as sexual selection, where really it's, even if some individual has an advantage in securing mates and produces more offspring, it might not be the result of sexual selection, some feature that they have that makes them, more selected for. It might be something cultural instead, and more of a random genetic change. So wild type, this is a term that is somewhat ambiguous. Sometimes it's used to refer to the dominant version of a particular gene. In this context and in this chapter, what we're going to use this term to represent is basically the ancestral state. So the wild type version of any gene, that allele is going to be what the ancestral trait was, what the ancestors of that population had. And so we can do some pretty neat stuff these days with statistics and modeling, where we can try to reconstruct the ancestral state from a sample of a population, and it actually provides some really helpful information on how that population has changed through time. Convergent evolution. Convergent evolution is when multiple populations develop basically the same phenotype, but through different mechanisms. And they can be shared phenotypic features that are not the result of common ancestry. And typically the best indication that they are not the result of common ancestry is that the genotypic strategy to accomplish that phenotype is different. And so we see some of these examples. The example we have in our textbook that we'll talk more about on Wednesday is lactase perseverance. So the wild type condition is probably to be lactose intolerant, to develop an intolerance to lactose after weaning. Although in different populations across the globe, there has been the development of a reliance on dairy products. And so you can see in those communities and in those people groups, oftentimes a genetic mechanism to ensure that lactase, the enzyme associated with breaking down lactose, is preserved after weaning. And basically, in every people group, the perseverance strategy, the genetic strategy to accomplish the same phenotype, is different, indicating convergent evolution and illustrating the microevolution in these groups. So, as you prepare for class on Wednesday, make sure you can explain these terms. If you have any questions on them, feel free to ask. During our class time on Wednesday, we're going to work through some specific examples of these mechanisms in human populations and illustrate how that has basically resulted in variation within the human genome.